Thank you, Michael, for the kind introduction. So as he mentioned, today my talk is on bispecific antibody therapeutics, challenges and opportunities and future directions. Today for the talk, I'm just going to check what's what here. Yeah, so that's a pointer, great. So I'm going to give a brief introduction of bispecific antibodies and then go into some of the mechanisms of action of common bispecific antibodies. And then I'm going to give you a little brief history lesson about the bispecific antibody development, and then talk a little bit about what commercialized bispecific antibodies and some of their modes of action. And then I'm going to go into a bit more of the meat of the talk, where I'm going to talk about the TurboChain platform at GenScript, and then discuss the cross-mAb technology and go through a few case studies. So as many of you will obviously know, monoclonal antibody versus bispecific antibody. For a monoclonal antibody, it only binds to one epitope. The first FDA-approved monoclonal antibody was actually called OKT3, and it was approved in 1986, where it was used for allograft organ rejection. Bispecific antibodies, however, are artificial proteins that combine to two different types of antigens, or two different epitopes on the same antigen. So kind of in a synergistic way, you can get binding of one part of the bispecific molecule to one antigen and improve the efficacy of that therapeutic. Saying that, there are very distinct challenges with bispecific antibody production, which I'm going to go into a bit more detail throughout this talk. So you can kind of think of bispecific antibodies as kind of like a monoclonal antibody cocktail. However, they're kind of the greater than the sum of their parts because they work in a more synergistic manner to elicit generally a better therapeutic output. And the aim for most bispecific antibodies are to treat cancer, as you will see soon. But there's also been some bispecific antibodies that have been implicated with chronic inflammation, autoimmune diseases, and also neurodegenerative diseases. If you look at the market trend, as you can see, like in 2022, the revenue was generated 5.7 billion, but this is predicted to go up compound annual growth rate of 24.1% up to over 220 billion pounds in 2032. So as you can see, this is a really fast growing market which therapeutic companies are looking to take advantage of. Go through a few of the mechanisms of action, because obviously as a bispecific, they can actually be more complex mechanisms of action compared to monoclonal antibodies. So one of the ways that they can affect is by this bridging mechanism. So the CD3 on the T cell can be bound to one arm of the bispecific, and then the other, the tumor-associated antigen, and the effector cell can then therefore kill the target cell. And this is generally the most common method of mechanism action for bispecifics. The next is kind of what we call in cis, where actually the bispecific molecule binds simultaneously to two separate antigens on either a tumor cell to prevent signaling or activation of those receptors to then enable signaling. Another way is to bring in a substrate and its enzyme into close proximity to then elicit an enzymatic reaction, for example, for the bispecific therapeutic that treats hemophilia A. Another method is for what we call piggybacking. So there are two kind of piggybacking mechanisms. One where you can actually cross a barrier, such as the blood-brain barrier, where you bind to a receptor, you get endocytosis, and then the other arm can bind to the particular target, which would normally be inaccessible. And the other way is for the binding of, say, a viral epitope, and then enable it to be then recognized by the neutrophil and engulfed. So in terms of the history of bispecific antibody development, the godfather was this guy called Nissenov, and he then demonstrated the first bispecific antibody concept and also looked at the first fragment-based bispecific antibody format. Hybridoma technology was then pioneered by Kohler and Meinstein in 1975. That enabled the production of these molecules. And also in 1983, the first asymmetric format was developed. In 1988, there was the invention of the SCFE fragment, and then followed that the first proposed solution to the light chain association issue through species-restricted light chain pairing. In 1996, there was the solution to the chain association issue through the use of complementary heavy chains, which we know as knob into holes. I'm going to talk a bit more about this in a bit more detail later. And then in 1998, the common light chain was used to tackle the light chain pairing problem. In 1999, there was the discovery of that the human IgG4 is actually a bispecific molecule. And then in 2007, the elucidation of this mechanism through the Fab-arm exchange process. Also in 2007, the DVD-Ig symmetric format was first pioneered. And then in 2011, cross-mAb technology was first developed by Genentech and Roche. And then I'm going to discuss the power of this technology in order to correctly fold these bispecific molecules. I'm going to discuss these later in the talk. And then in 2013, generation of bispecific IgG1 through Fab-arm exchange, and finally the first orthogonal Fab interface solution to the light chain association. So as you can see, we've come a long way in the last 60 years, and as you've seen throughout a lot of these talks today, that a lot more technology is now also being developed to enable the production of bispecific antibodies. So the first global approved drug was this mAbs, and this is a T-cell engager developed by a German company whose name actually I forget. And then in 2014, Blincyto as another T-cell engager, followed by this emicrelizumab. This is used to treat hemophilia A. And then, as you see, these are approved over the last from 15 years ago through. But as you can see, over the course of the last 2 years, we've had many more bispecific antibodies that have been approved. You can see in blue are the ones that have been FDA approved, and in black are by other approval bodies. But what this kind of really illustrates is that we are really ramping up our interest in bispecific antibodies and looking to actually further develop more candidates for the clinic. During the rest of the talk, I'm going to kind of go into a bit more detail about glofitamab production at GenScript, as well as this faricimab. So a little bit of an introduction to cross-mAb technology. So cross-mAb technology, as I mentioned before, was registered trademark by Genentech and Roche, and this was first proposed as a solution to the light chain association issue. So in terms of the cross-mAbs that have been in clinical trials, there are quite a number of them, and they have different formats from a 1-to-1 CHCl through to 2-to-1 and a 2-to-2, and yeah, I'll go into detail what these CH1, CL, and VL means. So what is the light chain association issue? So effectively, if you co-express two separate light chains and two separate heavy chains and let them assemble spontaneously, then you're likely to get up to 16 different combinations of these different light chain and heavy chain combinations, where only 12.5% of them are actually the product that you want. And actually, good luck trying to purify this 12.5% away from everything else. In order to kind of combat that, Roche developed this principle of domain crossover. So what that basically means is that they have this, for example, this heavy chain, which they have then fused one of the light chains to and used the other part of the heavy chain for the crossover, and they've left the other one unmodified. And in order to kind of glue these two together, they have engineered a bulky tryptophan residue into the part of the FC domain, and then they have engineered a cavity so that the first light chain can then interact neatly with the second heavy chain. So in terms of initially, the way to kind of get the light chains to actually correctly pair was to use this common light chain approach. However, this kind of requires de novo identification of the corresponding antibody pairs, which can be very challenging and time-consuming, and then also leads to the restricted availability and the diversity of the antibody. Another way is to prepare this Fab in tandem molecule, where you can actually fuse the two domains together. This cross-mAb technology is, as I mentioned, to either have this whole Fab component crossed over, or you can have the VHVL domain crossed over, or the CH1CL domain crossed over. And in terms of the production of these, for the CH1CL crossover, we're actually likely only to generate the correct molecule. So crystallography studies have shown that this is actually the case. In terms of the Fab crossover, there are 3 potential products that are observed. So you have this the correct product, but then you can also have heavy chain, and Fab pairing. In order to drive the production of the cross-mAb Fab, the idea is that you overexpress the Fab to kind of eliminate a lot of these monoclonal sort of heavy chain combinations. And then they can easily be purified away from the smaller Fab. In terms of the VHVL crossover, what potentially is made is this Benz-Jones-like mAb. But in order to kind of push the equilibrium over towards the correct product, then charge pairing is introduced. So you can introduce these positive amino acids for one of the Fabs to the negatively charged on the other one to ensure that the majority of the product is this charged cross-mAb. So now I just want to talk a little bit about our TurboCho platform. So in terms of our the way that we have been able to develop this platform, we follow this design, build, test, and learn method for each of these components at the top. So in terms of getting a highly productive CHO cell, we've undergone several rounds of cell engineering to ensure that the cell can be grown to a high density and producing and high production. Then we've also optimized the medium, the transfection reagents and the methods, as well as the vectors and the purification process. So all of this has enabled us to be able to express and from the gene synthesis all the way through to the production of human IgG, for example, in as little as 5 days. Our code and optimization platform is it has recently been upgraded from our GenSmart to GenSmart ZQ. So the GenSmart ZQ has enabled us to be able to improve the expression of poorly expressed proteins. So if you compare the expression of some of these human IgG1 antibody clones, some of them are very tricky to express using our GenSmart algorithm and GenSmart HQ, you can see that we've observed at least a tenfold expression relative to the original sort of algorithm. And when you kind of extend that across the human, mouse, and rabbit, you can see that we see vast improvements. In terms of our actual CHO cell, as I mentioned, this is GenScript's proprietary sort of technology where we've been able to improve the cell density, viability over various iterations of the engineering. So you can see we've been able to get at least a fourfold increase where the cells are at 90% viability. In terms of like the first case study, so I want to talk about this bispecific antibody format, this Fab-SC-FE-FC molecule, where we have a knob in whole and yeah, the Fab and SCFE. So we've expressed initially this in XB293F using a plasmid ratio of 1 to 1 to 1. And looking at binding to a protein A affinity resin, we only observed an SDS purity of 40% and a yield of 16 mg per litres. And as you can see from the gel, there are many different components within this purification. So we then used our TurboCHO platform, and we then also tweaked the plasmid ratio, so we have a 1 to 2 to 1 ratio, and then we also modified the purification strategy. We were able to obtain a purity of over 90% and a hugely improved yield. So it just goes to show the power of by optimizing the plasmid ratio, purification strategy, and using our proprietary cell line, we can actually greatly improve the yield and purity of the final product. The second case study I want to talk about is glofitamab. So glofitamab is actually a CD3, CD20 B-cell T-cell recruiter to tumor. So this was developed by Roche. This is actually a 2-to-1 orientation. And this is used to treat adult patients of diffuse large B-cell lymphoma, or DLBCL. So in terms of the sequence engineering, as I mentioned, this is a 2-to-1 sort of orientation with the VHVL format. So it has correct heavy chain association by knob in whole, and then the VHVL domain crossover, as well as the charge interactions to avoid Bentz-like Jones side products. So what we did was we coded and optimized the sequence of glofitamab, and then expressed a liter culture using our TurboCHO platform, and then we performed 2 purification steps. So first of all was protein A, and then followed by size exclusion chromatography. We were able to generate a yield of 24 milligram per litre with a very high purity and low endotox. So if you look at the diglycosylated mass, you see that we've got only the correct product, and the SEC shows a nice, beautiful single peak, and then also the CESDS is showing that it's very monodispace as well. The second case study is another cross-mab. So this has a CH1CL format and with a knob in whole, and yeah, so CH1CL1 ensured the correct light chain association. This molecule is a much tighter expression. So we only used a 100 mil culture using our codon optimization tool. And then in this way, we also used the protein A, followed by kappa XL. And as you can see, we were able to generate a very good yield with very high purity. And you can see the glycosylated mass shows that we obtained the correct product, and the sec shows it's nice and want to disperse. So to summarize what I've talked about, I've given a quick overview about bispecific antibodies and their mechanisms of action. I've gone through the history of the bispecific engineering, followed by a nice overview about some of the commercialized antibodies. I've talked about cross-mAb technology and shown you some case studies from what we've done at GenScript. If you want to know more information, you're welcome to scan this QR code, where you'll come to a page on our website where you can actually look at each of the different bispecific antibody formats that we've generated. This table is actually interactive, so you can see the sort of yields that you can expect, as well as the purification steps that we'd recommend to be done. And then I just wanted to kind of go through some other formats that we've made. So some SCFE Fab for bispecific antibody fragments, as well as SCFV2 Fab in either orientation, as well as a Fab VHH. And then we've also produced many different symmetric bispecific antibodies. And then also, as I've just kind of gone through, some asymmetric bispecific antibodies, including a different sort of purification method where we've done knob fermentation followed by whole fermentation, and then join them together, and more asymmetric bispecific antibodies. So yeah, thank you very much for your time, and I look forward to your questions.